DNA extraction. List of reagents: cell lysis solution, nuclear lysis solution, protein precipitation solution, 100% isopropanol, and 70% ethanol. Add 600 microliter cell lysis solution to a 1.5 ml microcentrifuge tube. Add 200 microliter whole blood to a 1.5 microliter centrifuge tube containing 600 microliter cell lysis solution. Mix well. Incubate for 5 minutes at room temperature and invert gently 3 times during the incubation. Centrifuge for 20 seconds at 14,000 rpm. Place the microcentrifuge by inserting a microcentrifuge tube opposite to each other. Set the microcentrifuge speed at 14,000 rpm. Press and hold the quick button for 20 seconds. Open the lid. White cell pellet will be visible. Remove as much supernatant as possible with micropipette, leaving behind the visible white cell pellet and about 5 to 10 microliter of residual liquid. Vortex the tube vigorously for 10 seconds to resuspend the white cell pellet in the residual liquid. Add 200 microliter nuclear lysis solution to the resuspended cell. Pipette up and down to lyse the cell. Add 67 microliter protein precipitation solution to the cell lysate. Vortex vigorously for 20 seconds. Centrifuge at 14,000 rpm for 1 minute. Open the lid and take out the microcentrifuge tube. The precipitated protein will form a tight, dark brown pellet. Prepare 200 microliter 100% isopropanol in a new 1.5 ml microcentrifuge tube. Pour the supernatant containing the DNA into a 1.5 ml microcentrifuge tube containing 200 microliter 100% isopropanol. Discard the precipitated protein pellet. Mix sample with inverting gently for multiple times. DNA will be visible like cotton thread. Centrifuge at 14,000 rpm for 1 minute. Open the lid, take out the microcentrifuge tube. The DNA will be visible as a small white pellet. Aspirate off supernatant and carefully lift the DNA pellet at the bottom of the tube. Add 200 microliter 70% ethanol into the DNA pellet. Invert the tube several times to wash the DNA pellet. Centrifuge at 14,000 rpm for 1 minute. Take out the microcentrifuge tube. Invert and drain the tube on a clean absorbent paper for 5 seconds. Add 66 microliter DNA hydration solution into the DNA pellet. Vortex for 5 seconds to mix the DNA hydration solution with the DNA pellet. Incubate the sample at 65 degrees for 5 minutes to accelerate rehydration. Take out the sample from the incubator. Measure the absorbance of the sample using spectrophotometer nanodrop. Make a blank by using 1 microliter DNA hydration solution. Put on the stage of the nanodrop. Click the blank icon to start measure the blank. Vortex the sample for 5 seconds and plus spin briefly to collect sample at the bottom of the tube. Take out 1 microliter of the sample and put on the stage of nanodrop. Put the name of sample ID, click measure icon to start measure the sample. Record the absorbance.